Good afternoon, everybody, and welcome to this Royal Society of South Africa lecture. I'm Stephanie Burton. I'm a member of the Royal Society Council, and it's my pleasure this afternoon to be hosting and to be introducing our speaker. Just a few little bits of information about the Royal Society before we start with the talk, and then I will introduce Professor Ramsay in a moment, but just so we can try and show you a little information about the Royal Society. Okay. There we go. We have had a number of little technical issues this afternoon, so apologies for that. But just to show you that we do have, we have a fairly big membership of the Royal Society, and of course being a member allows people to be part of these kind of talks, but also to have subscriptions to our journal, the transactions, and a number of other colloquia and workshops and so on that we host during the course of every year. So it's open to anybody, and you can find the application form to join the Royal Society on our website. I would like to welcome the three recently enrolled new members of the Royal Society, Dr. Rees, Professor Wasserman, and Professor Kaba. Welcome to you. And just to say that we are very pleased to see that we have members not only from university, but also from consulting companies. The Academy co-hosts these talks with us. So South Africa's National Science Academy, Academy of Science of South Africa, RSSAf, is a partner with us in hosting these talks and a number of other ventures, and our the website for RSSAf is also, of course, available to give you information. Just to let you know that, as I say, we have a number of these talks, and there are a number coming up in March and April, May and June, mostly online. Please do have a look at the Royal Society website, which has recently been rejuvenated and has a host of information on it. So please do take a look, and you will find information about our most recent talks and the ones that are coming up. We are recording this talk, and it will be available on our website afterwards. So now I'm going to introduce our speaker for today, Professor Michèle Ramsay, who is going to talk to us on the topic which you can see there, which is harnessing the power of genetic diversity in African populations. Professor Michèle Ramsay is a medical doctor, but also director of the Sydney Brenner Institute for Molecular Bioscience. She's a professor in human genetics and a South African research chair in genomics and bioinformatics at the University of the Witwatersrand. Professor Ramsay is also an expert and a well-known researcher in our country, well known for representing African science and in particular African populations in global science, and she actively promotes capacity building in the fields of genomics and precision medicine. She is co-chair of the International Health Cohorts Consortium and was a steering group member for developing the blueprint for a project for the African population cohorts, for the Academy of Science of South Africa's POPIA Committee, which is interesting because that's how we are working on a national framework for protecting personal information, and of course, closely linked to the ideas around genomics and genetics. So, as a member of the World Health Organization Technical Advisory Group for Genomics, Professor Ramsay contributes the lower middle income countries' perspectives to global genomics, promoting ethical, equitable, and fair practices and principles. So, Professor Ramsay, we're delighted that you're going to speak to us. I'm very much looking forward to hearing about all these amazing projects that you are doing. Thank you very much.
Yes, bear with me a moment as I try and get my slides up. I can see that it's just bringing the same one up. So I'll just close that a moment. There we go. Right, I think we're on the right talk now. Right. So, hello, everybody, and thank you so much, Stephanie, for that kind introduction. And just a little bit of a correction, I'm not a medical doctor. You know, I've been working in the field of human genetics for so many decades now that, you know, I've been interacting with doctors because I ran a diagnostic laboratory for human genetics for many years. But I stand before you as a scientist and somebody who really feels very strongly about human genetics and about African genetic diversity. So thank you so much for this opportunity to speak at the Royal Society meeting. And this is the first time that I'm visiting the observatory here, just noticing that, you know, they first had something at the site 202 years ago. So it's a great historical place to be at, and I'm very grateful to be here. So today I want to talk about how we can really think about the power of African genomic diversity. So human health is incredibly complex in so many different ways. And we know that it's not all about genetics, it's also about the environment. But genomic variation clearly plays an important role. And almost every condition of disease has a genetic component. And we also have to think about how the genome is regulated through epigenetics. And then, of course, not to forget the microbiome and the fact that this is important in terms of our metabolism and our immune response. But all of these things are mediators of the phenotype. And we occur somewhere on the spectrum from health to disease. And of course, that also changes as we get older. But I often think about health in Africa and the fact that we disproportionately have this burden of not only infectious diseases, but also non-communicable diseases. But there's no question that research has been dominated by infectious diseases. And what we're now seeing is this increased prevalence of non-communicable diseases. So it's really important that we understand the genomic components to these diseases and think about how, given our poor health infrastructure generally, we can actually use this information to tailor interventions. And here are some of the places that we work. So you can see that some of them are very rural and others quite urban. But I thought that I would start off just with the evolution of our species, because that has a direct bearing on genetic variation in Africa. And I want you to think, you know, at the top here is a long time ago, right at the bottom is where we are at the present day. So about 2,800, 280,000 years ago, there was the first split in our hominin lineage from hunter-gatherers into other peoples. And then at about 220,000 years ago, we had a split between the Central African foragers and the rest of Africa. And then more recently, the split between West Africa and East and Southern Africa. And this is very important because we understand that, you know, these populations vary from one another in terms of their genetic attributes. And then only about 70, 80,000 years ago, did we have everybody else splitting off. And this is really important because in Africa, people have a lot more genetic variation than do people from elsewhere in the world. And what we can say also is that Africans within Africa differ from one another much more than do people from Asia and Europe. So when we look at this, which is a famous map that most of you have seen, this out of Africa migration, that when people migrated off the continent, only a subset of that variation left. So there's a lot more behind that we haven't yet studied in Africa. And this is important because when we think about Africa, you know, we think about political boundaries. And this is a study that was done to look at readiness for research in the field of genomics and precision medicine. And as you can see, South Africa and Egypt are the most advanced. And some of the other countries are now also developing more capacity. But when we think about African populations, they don't respect these political boundaries. Of course, people traversed boundaries. And you can see all these migrations through different periods of the history of Africa. And that is important now, because whatever we develop in terms of precision medicine in South Africa may not be applicable in West or East Africa or other parts of Africa. So I always like to think a little bit about our continent and the fact that we have 1.5 billion people living in Africa, and that makes up about 18% of the world population. And we're expected to be one of the continents with the fastest growing population, so that by the year 2050, we'll have about 2.5 billion people. And, you know, the other fact about Africa is that generally people are younger. But when we look at genomic research, which of course informs precision medicine, fewer than 1% of the participants in genomic studies actually come from Africa. And I think, you know, that is something that we must think about, and the fact that we are missing out on this tremendous diversity and what its impact may be on health and disease. So we do know that disease patterns, clinical presentation, responses to drugs vary by geographic region and ancestral background. And although people often think of this in the context of the world, it's also true in the context of Africa. Let's think, for example, about sickle cell anemia. So about 80% of the cases of sickle cell anemia occur in sub-Saharan Africa, and it affects 7 million people. That is astounding. So where is the research going to be done on sickle cell anemia? We know that a lot of research on the genetics of hearing loss has shown us that different genes are involved than those involved in hearing loss in European populations. We know in terms of breast cancer, there are drug response differences. Prostate cancer, a beautiful paper published last year, showed that studying Africa gives us new genetic markers for predisposition to prostate cancer. When we think about chronic kidney disease, this is a disease which is 5 times more common in Africa than elsewhere in the world. And this is partly genetic because these risk alleles are much more common in African populations. And the interesting story there is that they've been selected for because of a sleeping sickness. So, you know, what might give you selection for some genes that protect you against something may also predispose you to other things. And then we also have variable response to beta agonists in people who have asthma. So I always feel so privileged to be part of these international studies and studies across the African continent, because I'm excited about this African Population Cohorts Consortium, which aims to develop population cohorts in different parts of the continent. And that is just now gaining traction. And I think what we really need in Africa are longitudinal cohorts where we follow individuals and understand not only their genetics, but also their progression through life. And then another very important initiative is the EU-Africa Personalized Medicine Consortium, which now has an action plan. And the South African Medical Research Council was very much involved in this 4-year project, which we now hope to take forward. So when we think about a traditional approach to prescribing medications, we have a group of patients with a similar disorder, in this case, colon cancer, and they all get the same drugs. But we know that some of them will react well, others won't have any effect, and some of them will have horrible adverse outcomes. And this is not very expedient. So what we would like in terms of a precision medicine approach is that people are treated in a way that tells us what the best outcome will be in the shortest possible time. So that is what we aim to do in precision medicine. And I just want to give you one example of where this comes into play. And this is for breast cancer. So it is well known that breast cancer is one of the most common cancers amongst women, but also that African women often have a much worse outcome than women elsewhere in the world. And the common treatment for estrogen receptor positive cancers is tamoxifen, which is then converted to the active component in doxifen. And what the research has shown us is that in Zimbabwe and South Africa, almost half of the people or the women who get this treatment do not reach therapeutic levels of endoxifen, which means that it has no effect for them in terms of their breast cancer. So we now know that there's a common variant in the CYP2D6 gene called STAR17, which occurs in about 30% of some African populations. And again, it varies across the continent. And these individuals are intermediate metabolizers, don't reach their therapeutic effect and will not respond to the drug. And there's a very simple solution. All you have to do is double the dose of the drug so that they attain therapeutic levels, and then that will address and hopefully combat their cancer. So, very simple intervention through research and understanding what is happening. I did tell you about this paper on prostate cancer in African men. And what you can see here is that the number of cases and controls are relatively modest, about 4,000 of each. And this was a study done in 5 different African countries. And what we know is that we have a similar performance to multi-ethnic and European studies with much larger sample sizes. So when we work in Africa, even with smaller sample sizes, we can discover extraordinary things. And I'm not going to go through some of those conclusions, but I certainly found them fascinating and important, because what they also show us are differences across these different African countries in terms of the genetic predisposition to prostate cancer. So I'd like to tell you a little bit about this consortium called the Human Heredity and Health in Africa Consortium, which has now sort of come to an end, although we are talking about sustainability. But it started in 2012, and this was a very ambitious project funded by the National Institutes of Health and the Wellcome Trust, with the aim of facilitating genomic research across Africa and thinking about how this genomic research can actually improve the health of populations in Africa. So one of the things that we did as part of this consortium was to sequence a lot of whole human genomes to look at diversity. And we were delighted to have a cover of Nature for that particular paper in 2020. And what you can see on the map on the left are the sites where we collected samples. And on the right is a principal component analysis, which is using genetic variation, hundreds of thousands of genetic variants, and reducing that to 2 dimensions. And each symbol is an individual. And of course, those who are closer together means that they are genetically closer together. And what you can see is that the populations definitely cluster in little sort of clusters together, and that those in West Africa are more similar to one another. And you almost see this climb across Africa where you go from west to central to south. So you can see the more people you add into your studies, the more you see a continuum, because of course, neighboring populations admix. So you would expect to see that. And East Africa, of course, is very different because of its history and its influences from many other parts of the world. So this is a study that we ran under the H3 Africa consortium called ABIGEN, and that stands for the Africa WITS In-Depth Partnership for Genomic Studies. And we did this study because we wanted to understand the genetic and environmental risk factors that predispose older adults in Africa to cardiometabolic diseases. So we started the study wanting to include 12,000 participants from all these 6 sites that we're showing here. So we had 2 sites in West Africa, 1 in East Africa, and 3 in South Africa. And what was really important to us was the whole process of the research. It wasn't about, just going and taking samples, coming out, doing all the research in one place and publishing the papers. It was about building a community. And it was a community of researchers that we built, but each one of those 6 groups actually went back to their communities to explain to them what this was all about. So that was incredibly important to us before we could even collect samples. So you can see why this has been a 12-year project. But it is about, sort of generating new knowledge at the end of the day and communicating in that in a way that we're not inflating expectation, because people often think genetics has the answers. But of course, it doesn't most of the time. It's very complex, and you have gene-environment interactions. But the reason we spend so much time talking to the communities is that we want to continue studying them. And it's also important for them to understand whether there would be some benefit to them in the long run, or in some ways in the more immediate time. So these are our 6 sites, and you can see 4 of them are very rural and 2 are highly urban, the one in Nairobi and the one in Soweto. So the Abujan study is really very complex. And, as I mentioned before, this has been going on for quite a long time. And one of the really special things about Abujan is that we've gone back for a second wave of data collection. So we have longitudinal data. So we could actually see when people progressed from not having a particular disease to developing it later on. So we started at baseline with individuals between the ages of 40 and 60, and then went back about 5 years later. And this is important in terms of the data that I'm going to show you. And what you can see there is that we retained about 60% of our cohort in the second wave. And this was really challenging because the second wave occurred during the COVID times. So I just want to acknowledge, all these amazing people we've been working with and, the different funders that were involved. But this is what we did. We collected a lot of data on different things, because remember, we were interested in genetic and environmental components to cardiometabolic disease. So it was really important for us to, obviously measure people, because we wanted to know about obesity. It was important to look at their blood pressure, because how many of them have hypertension. And then we did a whole lot of biomarkers from both blood and urine. But we also collected information on environment and lifestyle and on cardiometabolic disease outcomes, as you can see there. So here are just some pictures of us doing training, making sure that the informed consents were appropriate, that people understood what this study was all about. And, we had different layers of engagement. First of all, the chiefs, if it was a rural area, then communities, then small groups, as you see on the left here, to whom we explained what was going on. And then ultimately a one-on-one where people had an opportunity to ask questions about the project. And then collecting blood samples, which some of our partners hadn't done before, but did because of this project. And this has now opened up possibilities for them going forward with other research. So I want to show you these data on obesity across Abujan. And first of all, these are the 3 South African sites. And I want you to look at the F for female, M for male. And what you can see immediately is that women are more likely to be obese than are men. But this isn't true everywhere. That's not true in West Africa. And what you can see is that there were small differences from wave 1 to wave 2, 5 years later. But I wanted to also show you some of the data on the cardiometabolic disease outcomes. And I've just chosen diabetes and chronic kidney disease here because they tell us different things. So what you can see is when we look at prevalence on the y-axis as percentage of the population, and wave 1 of data collection with the closed circles, wave 2 with the open squares, what you can see in South Africa is that the prevalence of diabetes almost doubled in 5 years. And we've just had our paper accepted in Lancet Global Health, which actually talks about the incidence of diabetes. And you can see how important it is here to think about interventions. And what we've done is to look at the risk factors for people developing diabetes in 5 years. And what you can see in terms of chronic kidney disease is that that has also increased dramatically so in 2 of our sites, but not the 3rd South African site. And when you look at the 2 West African sites, what you can see is that kidney disease has increased, but diabetes has not. So by doing this multi-country, multi-regional study, we can discover a lot, and that is very important. This is just showing you genetically how different these populations are, and this, similar to what I showed you before. But you can see that even within AbiGen, we have a lot of population structure. And this complicates the way in which we do the genetic analysis. Because remember, I was saying that, the same interventions are not going to work everywhere in Africa. So we have to adjust for these changes in genetics. So now I'd like to just briefly take you through 3 projects that we've done in the AbiGen study. So the first one is looking at genetic associations with complex diseases. And I don't expect you to take all of this in, but just, to think that this is an army of people who actually did this work. And this particular study was looking at the genetics and genetic associations with lipid traits. And we know that, high cholesterol increases your chances of having a stroke and of other cardiovascular diseases. And what we found in our association study is that we often replicated genetic variants that were already known in other populations, mostly European populations. But sometimes we would find the same gene, but a different variant in African populations because they are different. And this means that that particular gene is biologically important. But every now and then we find something completely new. And in the red block there is a new locus that had never been found, despite people having millions of participants in the European studies. So this is an African signal that we need to understand better and understand how it contributes to cholesterol. For those of you who follow genetics and maybe, your own health, you think about, how could you know very early on in your life whether you have an increased susceptibility for a particular disease, an increased genetic susceptibility? And very often, family history will tell you that. So if there's a family history of type 2 diabetes, it means that your risk is higher. But wouldn't it be interesting to be able to look at your genetics to see whether your risk is higher or not? Because, even 2 siblings of a parent who has type 2 diabetes may have very different genetic risks. Okay, so what we can see in this is deciles of polygenic risk scores, and then on the y-axis sort of saying how predictive it is. So it's not very predictive because that is %. But what we can see is if we build our models on multi-ancestry data, they perform better than if we just build our models for polygenic risk scores on European populations. And the reason it's not that much better for African populations is that we just don't have a lot of data. So we need to think now, when we want to predict somebody's risk of developing disease, whether we only want to look at genetics or we also want to look at other things. And this is a beautiful study that was done by a PhD student, Michèle, and she did what we call an integrated risk score, where she looked at genetic risk, but she also included non-genetic risk and came up with a model to look at your combined risk for developing cardiovascular disease traits. So the traits that she looked at were dyslipidemia, hypertension, and obesity. And the non-genetic factors that were taken into consideration were smoking, alcohol, diet, exercise, and sleep. And what you can see when you look at those graphs, the red is the genetic prediction. The blue is the non-genetic prediction. So you can see non-genetic things contribute more to your risk for these conditions. But if you combine the two, you get a better prediction, which is the green. So I'm just very quickly going to transition now to something else, and that is that we don't have a lot of African data, and therefore we don't understand the interpretation of the human genome from Africans very well. So one of the things we wanted to do was understand regulatory variants in the human genome of people from Africa, because most of the data we have is based on Europeans. So we partnered with a company called Variant Bio to do this particular study. And it was also nested within AbiGen and H3 Africa. So what we did was that we collected blood samples from about 750 individuals. And we did the study only in South Africa because it was during COVID and it was just too difficult to do it in our other AbiGen sites. And what we did was do whole genome sequencing together with whole transcriptome sequencing, which is the messenger RNA. to try and see if we could identify genetic variants that influence the levels of gene expression. And this is very important when you're trying to interpret sort of variants in the human genome. Could I ask the technical person, please, to plug in my computer? I can see my battery is going to go off in a minute. Thank you so much. And just again to say, because this is South Africa, it may not equally apply in other parts of Africa. So very important that we actually generate data in different places of Africa, not only in South Africa, of course. And through bioinformatics, you can actually look at the blood and the transcriptome in the blood, and you can do what we call cellular deconvolution. And we're just going to plug this in a minute. Do you need something else? There we go. And what is absolutely extraordinary is that they can identify the relative proportions of 40 different traits. So I was just telling you about the complexity of the blood and the deconvolution into 40 different relevant blood types. And what we did then was to also try and understand, do we need to put the picture on again? Let me just do that so they can see me again. There we are. And right, so what we wanted to do was see whether there was an association with particular diseases and certain enrichment of cell types. So I won't go into that in any detail, but just to say that in these 750 South African samples, we found on average 4.3 variants per gene, whereas in European studies, they only found 1.7. So I think the only point that I want to make with this slide is that whenever you study African genomes, you will find new things and you will discover a lot more variation. And that is really important when we think about associations with disease. And we now have called this resource SABRE, which stands for the South African Map of Blood Regulatory Variation. And that has now been accepted for publication in Nature Genetics. And we have the whole benefit sharing that we did together with Variant Bio also in a manuscript that we hope will soon be published. But all these data are available to people in the world to use. And I think that is really important, that as South Africans and Africans, we really have to think about how we share our data responsibly so that it can have global benefit. This is the last example that I want to give you, and this is about the microbiome. I'm sure all of you have heard about the microbiome and try to understand its effect on your health or whether it's related to the diseases that you have. And what you can see here is all the different things that the microbiome actually affects. And this incredible group of scientists based at the Sydney Brenner Institute in Johannesburg at Wits, but also at Stanford University, have now published their paper, which we're also very thrilled to say has been accepted in Nature. So this is about collecting stool samples, because this is the gut microbiome, extracting the DNA, reading the sequences and putting them together and calculating the abundance of different microorganisms. So this was also nested within Awigen, so you can see the same centers involved. And what we can show is that rural communities tend to have much more diverse microbiomes than do urban communities. And the more diverse your microbiome is, generally the healthier you are. But we also did studies to look at associations between the microbiome and HIV. And what we can see is that individuals who are HIV positive, even if they are well controlled through antiretrovirals, have a less complex microbiome. And even through antiretrovirals, their microbiomes haven't recovered. So this is very important to also understand how that might affect their health. So there's a wealth of information in this particular paper. And what we're really excited about is that we got the cover of Nature showing in these three biomats some real science. So those are the phylogenies of the species of microbiomes from this particular study. So as you can see, it will be published in a couple of days. So I want us to think a little bit about the advantages and approaches to genomic studies. And, you know, when we do these genomic studies, we need to be mindful of many different things. When we're studying diseases, we need to be very careful about the phenotypes, because we know that there are differences in clinical manifestations of certain common diseases like kidney disease, for example. Even things like, you know, chronic kidney disease is different because in Africans, it's usually associated with hypertension, whereas in European populations, often with diabetes. So we need to understand what the differences are. We also need to know that it's different in different regions of Africa. And we need to think very carefully about how we collect data, and that we need to be very careful about the consents and the ethics so that we can actually share that data and make it available to others. But we're very interested in this whole issue of transferability. Like, if we do this work in South Africa, will it apply in East Africa or elsewhere? So I think, you know, the message is we really need to do work across different places in Africa, because, as I said right at the beginning, people in Africa are so different. We need to take that into account. And it's not only about the genetics, it's also about the environment. And then, of course, what we would love to do is bring that to full circle where it can actually have a benefit in terms of the clinic. So Stephanie said right at the beginning that I was part of the a group under the WHO, which was looking at responsible sharing of data. And it all started because one of the first reports of the WHO Science Council was actually this one, about accelerating access to genomics for global health, and really saying nobody should be left behind. So this has resulted in a guidance document thinking about, you know, human genomic data in terms of how it's collected, how it's accessed, how it's used, and how it's shared. And there were 7 principles that came out, and Kira Staunton was involved in this, and lovely to see you in the audience, Kira. But it was really an international group who debated this for many months about what would be acceptable across the world. And this is, again, where because I've worked in South Africa and different parts of Africa, I could bring the experience and see whether these principles would equally apply in different parts of Africa. So you can see the emphasis here really on the people, the communities, and how they need to be informed and given information about what it means. So there also needs to be mindfulness of access to benefit, about collaboration and partnership, responsible stewardship, but also accountability. And, you know, this is going to be also debated at a UGO conference in March in Durban, where we think a little bit more about, you know, how do we think about genomic data in different countries in different parts of the world, and how can we share it for the benefit of everybody? So I hope that I've shown you a little bit that, you know, harnessing the power of genetic diversity in African populations is incredibly important, and it will help us address some of the complexities in health across this whole spectrum. And right at the end, saying that it's really a global imperative, because whatever we find in African populations is likely to be able to benefit people anywhere in the world. Because, as I said, there's only a subset of African variation that left the continent, but a lot of it is still here, and we need to explore it, and we can find novel approaches to health. So I think with that, I will just finish, and happy to take some questions.
Michèle, thank you for an absolutely so much information, obviously so much data, and I'm sure everybody is completely bowled over by how you've made it sound simple, but I know that it's very complicated. So we can ask some questions and answer any. I would suggest that if people are online and would like to ask questions or make a comment, that you try to use your Zoom hand. It's not always all that easy, but if anybody online would like to ask a question or make a comment, please do go ahead through the Zoom system. And in the meantime, would anybody in the audience like to ask a question? Please go ahead. If you could just introduce yourself.
Hi everybody, it's my name, Rangani. I am from Stellenbosch University. Thanks so much, Michèle, for a wonderful presentation. I work a lot on plant genomes and microbiomes and how plants change and stress. Lovely presentation. I think one of the things you highlighted was this immense genetic diversity that exists within African populations. Similarly, in plant studies, we see the same things. Certain locations have different levels of diversity. Ultimately, the goal is to end HIV prevention, end world hunger. I keep coming back to the same point, is that no two biological samples are the same. Any scientist. I just can't figure out how do we effectively study these immensely diverse systems to come up with solutions that are specifically tailored for that population, or maybe even we can translate it to other populations, subpopulations, because we end up with a solution that was created in Europe. And I can give an example where a lot of microbiome intervention studies in agriculture have been focused on the addition of a specific microbe in wheat or some field in Netherlands, and it showed that the plants grew better, but it doesn't really work in Africa. So we have this data that everything is different in Africa, everyone is different, and then what? How do we move forward from we're all different?
Timiling, thank you so much for raising that, because it's so true. You know, when you look at human populations, not one of us is the same as somebody else, except if you may be identical twins, and even then you'll have some differences. But, I think your point is well taken. And, we need to, obviously people who are genetically more similar will behave more similarly, but it also depends on their environment, because there are beautiful studies that have been done of Ghanaians in the Netherlands and the UK compared to Ghanaians in Ghana. And the prevalence of kidney disease is very different. And it's not because their genetics is different, it's because the environment is different. So I think, the take-home message, and, I love this whole thing about One Health, talking about agricultural, environmental, and human health. And there's a real move towards that, and we need to understand all those connections. But if we don't do the research locally and in Africa and in different parts of Africa, we're not going to find solutions that are appropriate for us. So, I'm really pleased that you are working, in this field in agriculture, because I think, if we don't look, we don't know. And we're not going to, this is the whole thing about transferability, is that you cannot take solutions developed in Europe and bring them to Africa. We found that even with monogenic diseases in African populations, for instance, cystic fibrosis has different mutations in African individuals. So a test that was developed in Europe is not going to work if you apply it here, because you're going to say, no, they don't have it, but you looked in the wrong place. So you need to do the research to really understand what you need to do and how you would intervene or how you would use it. So the same with the microbiomes around the plants and whether they're going to thrive or not, given a certain diversity of microorganisms.
Thank you. I see Leslie has her hand up. Leslie, please go ahead. Don't forget to open up your mic here.
Thank you. Thank you for a totally amazing talk, Michèle. I'm blown away. And you must forgive me if my questions are totally stupid, because I'm not part of this. This isn't my discipline. So firstly, I probably got the name wrong, but the Khoi split off much earlier. Did you ever have any of their descendants in your study, because they would be that much different? And the second one is, where do we get our gut biota from? We don't inherit it, do we? Presumably it comes from what we put in our mouth, although we can put different things in our mouths. Like, I know elephant calves eat the dung of their mothers, so they get some of the right things. Thank you. That's me.
Thank you so much, Leslie, for both of those questions. So maybe to tackle the first one about the Khoisan, and you're quite right that they split off so much earlier. So we're going to see much more and very different genetic diversity amongst them. But we've done a very interesting study in the 3 different sites in South Africa where we've worked, and we've actually looked at the Khoisan admixture in those 3 groups and showing that some of them have up to, maybe 15 to 20%, and others only have 4%. So we're hoping through our studies to be able to tease out, they often call about, what do they call it? And regional ancestry, where you can look at parts of the chromosome and understand whether they are more likely to be of Khoisan origin than other origins. And so you can start looking at associations. So Leslie, we are looking at that, and we find it absolutely fascinating. And we're doing a new study now where we're doing 1,000 whole human genomes from 9 groups that are not represented. And some of those are Khoisan populations, but in Zimbabwe and Kenya. And so, I think we're going to discover a lot more variation that we don't know about yet. So lots to be done and to be understood. Your second question about the microbiome. So I have to say, I'm not an expert, but I know that, some of the microbiome you get during birth through the birth canal from your mother. But you also do get it from your environment. And we know that when people take antibiotics, for example, they reduce the diversity of their microbiomes, because that's the whole purpose of taking the antibiotics. But you kind of hope you don't kill the good bugs, but only the bad ones. But I think, it is an opportunity because you can modulate the microbiome, and it's also very much influenced by diet. And what we've seen in our study across these 6 different regions in Africa is that the similarities are more according to geography than anything. Because, we see these rural-urban differences, but we also see geographic differences. So there's so many things that influence your microbiome, and it is changeable. So you can actually, improve it. And in some instances where people have very serious diseases, working on the microbiome can really make a big difference. So I'm just hoping that people will watch that space.
It is absolutely fascinating. Just to ask one simple question before we finish. Africa stands to some extent alone or outside of or different from the rest of the world. Have any studies like this been done in other regions? This is long term, and it's not one study. I really appreciate that. But would there be any possibility of comparing what you find with Africa with what you find in another region of the world? China, for example.
Yeah, so more and more countries are now doing what they call national genome projects. And they're giving us a lot of information because we have these big, what they call almost like biobank projects. So in the UK, they've done 500,000 individuals. In the US, they're now doing a project called All of Us, which is like 1 million people from the US focusing on understudied groups. But there's a lot of work going on across the world. And what we do want to do is global comparisons. But whenever you look at genetics, there's a lot less genetic diversity anywhere else when compared to what we see in Africa. Interestingly enough, in Brazil, they've done really interesting work because Brazil has such an interesting population because it has, American populations, but a mixture with Europeans, but also with Africans. And there's some communities there that are very African in terms of their genetics, because there's one community where there were slaves who ran away and formed a new community that is almost entirely African in terms of their ancestry, but they're living in Latin America. So these studies are fascinating and ongoing in different parts of the world.
Absolutely amazing. So colleagues and guests who are online, I think we are more or less at the end of our discussion. I'm quite sure we could continue asking questions for a long time, but I think at this stage, I want to particularly say thank you to Michèle. One just does not realize the wealth of information and the richness of this research, and the fact that you have so many publications in Nature speaks for itself. It tells us that this is the kind of work that is really important for the world. So, Michèle, thank you very much for an absolutely wonderful, wonderful talk. And yes, long may it continue. And I think we should ask you to come back in about a year's time to tell us a whole lot more about what you found in the meantime. I see some of the audience also applauding. Thank you very much indeed.